Hi everyone, this is Sandesh D from St. Philomena's College, second year BSc from the Department of Botany. Today I am going to discuss with you the ABC model of flora organs in Arabidopsis thaliana. Let's see first what is this Arabidopsis thaliana. It is a plant which is well used by the scientists to study the concept of ABC floral development in plants. Arabidopsis thaliana is a small flowering plant commonly known as tail cress. It belongs to the Brassiaceae family, which includes mustard seed and cabbage. This plant is widely used in scientific research, particularly in molecular and genetic studies, due to its simple genome and short life cycle. It is a well-known plant which is used by all the scientists for the easy understanding of the concepts, especially with regard to this ABC model of flower development. And this is the image that we see of the plant, and this flowering plant is what is known as Arabidopsis thaliana. Now let's look at the characteristics of this plant. The size. What is the size of this plant? It grows to the height of 10 to 20 centimeters. It's just a small plant. Leaves. Roseate arrangement with small serrated leaves. Flowers. The color of these flowers are white to pale purple flowers with 4 petalets. Life cycle. Short life cycle, completing its life cycle in about 6 weeks. So it has a very short duration of lifespan. And genome, fully sequenced genome, aiding genetic studies. Arabidopsis thaliana serves as a model organism, providing valuable insights into plant biology development and responses to environmental stimuli. Now let us study the ABC model of floral organs in relation to the plant Arabidopsis thaliana. The first thing that we need to know is the meristem. The meristem is a part of a plant which grows and develops, and meristem grows and develops into a floral meristem, and the ultimate product is the flower. And ABC class of homeotic genes. Let's see what these homeotic genes means. Homeotic genes are that which govern the development of plants. So homeotic genes are that which govern the development of plants. And what do you mean by ABC class? Genes work together in the development of plants. These are ABC are nothing but a cluster of genes working together in the process of development of flower. The same meristem gives rise to floral meristem, and the end product is the flower. Now let us see what the meristem consists of. We are all aware that meristem tissue consists of 3 types of meristem. There is apical meristem, lateral meristem, intercalary meristem. The apical meristem gives rise to 2 types of meristems again, SAM and RAM. SAM is the shoot apical meristem and RAM is the root apical meristem. Once again, the shoot apical meristem gives rise to floral meristem. Floral meristem gives rise to the ABC class of homeotic gene arises. So we have seen here the meristem giving rise to floral meristem, and the product is the flower. In the process of meristem giving rise to floral meristem involves this process wherein the meristem, that is the apical meristem, gives rise to two types, SAM and RAM, and from the SAM, that is shoot apical meristem, originates the floral meristem, and ABC class of homoetic genes gives rise to the flower. Now let us look at the flower and study its part. The first thing we see here is a stamen. Stamen is the male reproductive organ of a flower. It's also known as androecium. And then we have corolla. Corolla is nothing but petal. And then we have carpel. Carpel is the female reproductive organ of the flower, and it's known as gynoecium. And then the last part of the flower we have sepal, which is also called calyx. Now let's look at the constructive floral diagram. How the flower is developed layer by layer. The first thing we see here is a circle. The first layer of the flower is sepal. It can be called as a whorl. The first whorl of the flower is sepal. And then the second whorl is petal. And the third whorl is stamen. And the last final one is carpel. Carpel is nothing but the female reproductive organ, which can be also called as gynoecium, and stamen is nothing but the androecium, the female reproductive organ, then petal and sepal. Now let's look at the homoetic genes for flower development. We have already seen what these homoetic genes mean. Now let's look at what these homoetic genes have got to play a role in regard to the development of flower organs. Homoetic genes are divided into three parts: gene A, gene B, and gene C. So gene A gives rise to AP1, which is apical 1, and AP2, apical 2. So A gene is responsible for two different AP1 and AP2. And then we have B gene, which is responsible for AP3 and PI, that is pistillata. And then gene C gives rise to agamus. Now let's look at this A gene first. A gene gives rise to AP1 and AP2. And AP1 and AP2, that is apical 1 and apical 2, has a great role in relation to the production of sepal and petal. Then coming over to the B gene, which gives rise to AP3, that is apical 3, and PI, that is pistillata. B gene has a great role in production of petal and stamen. And then we have C gene, that is AG, agamus. It gives rise to carpel and stamen. Now let's look at them in still more deeper. Let's look at the constructive floral diagram that we already see. The outermost whole of the flower is known as sepal, and the layer after sepal comes petal, and then comes stamen, and then the innermost is carpel. Now let's look at the generalized gene interaction diagram. We see here gene A, gene B, and gene C. And now we are going to see what this gene A, gene B, and gene C got to do, and what is their role in relation to the production of these four elements or four whorls of the flower. Gene A gives rise to sepal. That is, gene A alone gives rise to sepal. And then gene A along with gene B gives rise to petal. Both gene A and gene B has a role to be played in the production of petal. Then coming over to stamen. Stamen is produced when both gene B and gene C interact each other. So Stamen is produced when there is gene B and gene C is present. And then we have carpel. Carpel is produced only with the gene C. And this is generalized gene interaction. We also see underneath A and C mutually antagonistic. B gene does not express individually. When gene B does not express itself individually, whereas gene A and gene C express individually. When GA expresses individually, it gives rise to sepal, and GC expresses, it gives rise to carpel. Now let's look at the mutations. We have when gene A expresses individually, we have sepal. When gene A and gene B express together, they produce petal. And then when gene C and B express, they give rise to stamen and carpel. Now let's look at, we have already seen this diagram. Now let's look at the mutation of A gene. What happens when A gene is not present? As we have already seen before, when A gene is absent, there is an expression of C gene. In the place of A gene, the C gene is expressed. And so we have, in the first word, we have carpel. And when gene C and B interact each other, they give rise to stamen. And then once again, when gene C and B interact each other, they give rise to stamen. And gene C alone gives rise to carpel. And this is the result when there is a mutation with regard to gene A. Now let's look at what happens when there is a mutation with regard to B gene. What happens when the B gene is absent? We see A gene alone produces sepal. And once again, A gene alone produces sepal. And gene C alone produces carpel. And once again, gene C alone produces carpel. There are always 4 whorls in a flower. And since B gene is absent here, we see the 2 sepal whorls and 2 carpel whorls. And this is the result when there is a mutation with regard to B gene. Now let's look at what happens when there is mutation with regard to C gene. When there is mutation with regard to C gene, there is the presence of A gene. In the place of C gene, the A gene acts. So A gene alone gives rise to sepal. When A gene and B gene are present, they give rise to petal. And once again, when A gene and B gene are present, they give rise to petal. And once again, when A gene alone is present, it gives sepal. So we have sepal, petal, petal, sepal. As we are already told that every flower will have 4 whorls, and here with regard to the mutation of C gene, there is the first whorl is sepal, second one is petal, third one is petal, and the last one is sepal. And this brings us to the end of a study in relation to ABC model of flower organs development with regard to Arabidopsis thaliana. Thank you. See you once again.